On the left, a tumor cell aggressively multiplying out of control. On the right, a neuron rapidly withering away. Standard medical models treat these as isolated events, driven by unrelated errors in the genetic code. But looking closely at the cellular mechanics reveals a paradox. Cancer involves a system refusing its scheduled death, while Alzheimer's involves a system executing that death sequence prematurely. Thermodynamic profiling shows these pathologies following a distinct symmetry, moving in opposite directions along a single mathematical axis. Classical bioinformatics pipelines routinely miss this relationship because they slice proteins into truncated fragments to save computational processing power. To see the full picture, we have to abandon the linear reading of DNA sequences and look at the cell through a biophysical lens. Moving beyond the physical structure of the double helix, we enter a space where disease operates as a series of informational phase transitions. This requires a new computational framework, designated as the consciousness engine, built to map the entire biophysical space of a living cell. This engine calculates the genetic sequence alongside environmental physics, specifically mapping how intracellular pH and oxidative stress dictate the behavior of the genome. Within this matrix exists a cellular kill switch, a strict thermodynamic threshold that ultimately determines whether a cell survives or undergoes apoptosis. Disease is the result of environmental pressures forcing an unnatural inversion of the cell's natural phase state. To calculate these phase states, the model upgrades classical sequence alignment to an 18-qubit quantum architecture. Assigning to first 14 qubits maps the primary peptide chain. The model adds a toggle qubit for the wild-type and mutated superposition state. 2 more attach for external pathogens, and the final qubit maps isotopic variance and nuclear mass shifts. These final 4 additions are the quantum control parameters. They encode the physical and chemical environment directly onto the peptide. The completed 18-qubit vector serves as a high-fidelity map of the host's interaction with its pathogens. By treating environmental triggers as computable variables, the model calculates the exact coordinates required for cell survival. This 18-qubit array organizes into a 12-branch tree tensor network, capturing nonlinear dependencies between sequences and acting forces. These pathways then compress into a rigid 2x3x2 geometric tensor, isolating every possible combination of discrete disease states. To evaluate the stability of this matrix, we use quantum Shannon entropy, a metric that calculates the system's total information capacity. The engine reveals that the target biological system operates at exactly 8.24 bits of entropy, well below its maximum potential of 18 bits. Operating at 8.24 bits indicates a highly constrained, pinned physical state, where the cell has extremely limited degrees of freedom. This constraint forms the thermodynamic threshold where the cell initiates either survival or apoptosis. Severe pathologies emerge only when this specific entropy threshold is chemically breached or artificially masked. The physical controls for this threshold are intracellular pH and oxidative stress. They operate as environmental logic gates. An alkaline pH logic gate induces constructive interference in the quantum waveform. This masks the mutation, locking the cell into a zero state, blocking apoptosis. Conversely, an acidic environment forces a 180-degree phase flip, causing massive destructive interference characteristic of Alzheimer's. These environmental logic gates actively decide the cellular trajectory, forcing the system into either an unnatural state of survival or a premature death cycle. We can observe these phase states driving actual molecular chemistry by examining TP53, the gene responsible for tumor suppression. This diagram illustrates the TP53 sequence, with concentrated CpG methylation hotspots between positions 19 and 69. These precise coordinates act as phase stabilizers. Under high oxidative stress, the chemistry breaks down. Methylated cytosine molecules at these hotspots spontaneously deaminate into thymine. When mapped back to the quantum model, this chemical transformation represents a permanent physical bit flip in the genome. This flipped bit silences the tumor suppressor signal, ensuring the collapse of the DNA's structural integrity. Localized structural collapses, triggered by environmental stress forcing a bit flip, Define the vulnerability of these genetic sites. This thermodynamic system extends upward, interacting directly with the brain's neural oscillations. To map this, the model uses the theta matching system to achieve what is termed transtemporal unification of disease states. Look at these dual readouts. On the left, the high-frequency jittering beta and theta waves typical of Alzheimer's. On the right, the rigid, flat waveform of a cancer state. The consciousness engine mathematically superimposes these two opposing signals. Mapping these two signals reveals that the high-frequency jitter of neurodegeneration and the low-frequency rigidity of malignancy function as phase opposites. Superimposing them creates a point of destructive interference, mathematically stabilizing the system. This confirms that these disease states are ultimately informational timing errors, presenting as buffer overflows in DNA repeats or phase shifts in neural frequencies. The data shows that completely disparate mutations in genes like TP53, KRAS, and BRCA1 operate within a singular information thermodynamic system. This chart displaying Kappa-6 validation confirms the cross-analysis. The identical mathematical signature holds true across 14 distinct pathological conditions. Targeting specific mutations or pathogens ignores the broader informational logic gate that dictates cellular behavior. Medical treatment must evolve to phase-correct the cellular environment, repairing the underlying logic gates before the structural collapse occurs. By utilizing harmonic resonance and temporal matching, the engine aims to flip the biological bit back to a balanced homeostatic state. The interaction of physical forces, genomics, and consciousness reveals that treating disease relies entirely on retuning the human system.